Indeed, there are growing evidence in literature that RLS and inflammation have a bidirectional relationship, because RLS has a complex and multifactorial pathogenesis, and inflammation fit perfectly into this model because it could act on both sides, particularly increasing the environmental burden. And RLS is associated with inflammatory infection disease, and among them, the most common are the gastrointestinal ones. I think that a so complex pathogenesis requires the most complete approach to be understood, and so the omics represent promising techniques that could help us to find prognostic and diagnostic biomarker and therapeutic strategy with a personalized approach. And so, to the best of our knowledge, to date there are no metagenomic study in RLS, and so study that analyze the microbiota composition in RLS. And RLS is a sleep disorder with a pronounced circadianity, and other sleep disorders are associated with alteration in microbiota composition and also chronotypes. So our hypothesis is that gut microbiota could be one of the environmental risk factors that influence the RLS phenotypes and symptoms. In our pilot study, we try to understand if the gut microbiota composition could distinguish RLS patient from control, and if it could influence the RLS clinical phenotype. So we compared a population of 30 RLS patients with a control group of 29 healthy subjects matched for age and sex. So we collected the stool into sterilized tube, and then the microbial DNA was extracted, amplified, and sequenced according to the standard metagenomic analysis. And finally, the microbiota profile was determined by using the DATA2-PP line. We did not observe any differences in the general macro composition of the microbiota, so for example in the ratio Firmicutes bacteroidetes, but when we look at the genus, we observe a significant differences between the RLS group and control. In particular, there is this genus, the Lachnoclostridium, which is significantly reduced in RLS population. In particular, it correlates also with the clinical phenotype because it is significantly reduced in patients with a long disease duration more than 50 years and in the absence of sensory symptoms. We are really at the start of this trip because, as I said, to date there are no metagenomic study in RLS, but it could serve as a potential diagnostic biomarker because it could distinguish RLS patient from control. But we have to overcome our limitation. For example, in this current project, which is the Biomarker Inflammation RLS, which will be supported by the RLS scholarship, we want also to include a population, a control group of insomnia patients without RLS. And so this could be the key to observe if it's a real specific diagnostic biomarker. And then for the prognostic part, we want also to observe during a follow-up of at least 1 year the modification of the microbiota composition in RLS patients. Part of this objective, because it's a really complex project, so we want to start for sure with this involvement of the control group of insomnia patient, as I said, to confirm the specificity of this identified biomarker. But then we want to move to what we think that it's the real actor of this chain, the shorty chain fatty acid, because the Lachnoclostridium genus could produce these shorty chain fatty acid, which are able to regulate the integrity of the gut barrier, but at the same time they could cross the CNS, the blood-brain barrier, and influence the CNS homeostasis. So we want to also to perform on the RLS stool sample a metabolomic analysis in order to profile this shorty chain fatty acid. Finally, we want also to include other biomarker of inflammation, systemic inflammation, and also we want to correlate with the iron status in this patient in order to give a complex molecular signature of this patient.